Welcome to the Unraveling the Scriptures channel. The Israelites possess an ancestral DNA that traces back to ancient populations of the Middle East, sharing a Semitic origin like they do. In this video, which is the second part of our series on the genetics of the ancient Israelites, I will explore other Jewish groups that also carry this same Semitic heritage in their roots. Using a wide range of genetic articles, we will delve into the origins not only of the Israelites, but also of some well-known contemporary Jewish groups, revealing ancient secrets preserved in their DNA. Throughout this study, we will address how these genetic connections link today's Israelites with biblical patriarchs like Abraham and unravel intriguing mysteries about the migrations and mixtures that shape this people's identity over the millennia. The mysterious genetics of the ancient Israelites, the DNA of Abraham. Genetic studies of Jews are part of a fascinating field of population genetics and are essential for analyzing the migratory chronology of the Jewish people. These researches are accompanied by other fields of knowledge such as history, linguistics, archaeology, and even paleontology. All these disciplines converge in a joint effort to thoroughly investigate the diverse origins of different Jewish groups, also known as Jewish ethnic divisions. One of the greatest mysteries that these studies seek to clarify is whether there is a common genetic heritage among all Jewish branches, meaning whether there is a shared Semitic ancestor, like Abraham, that unites all these populations. For this, several studies have been conducted using three main types of genealogical DNA tests: autosomal, AT-DNA, mitochondrial, mtDNA, and Y chromosome, Y-DNA. Autosomal DNA tests, for example, aim to analyze the entire genetic composition of an individual, revealing the populations that form the so-called genetic isolates among Jewish groups. Mitochondrial DNA, mtDNA, examines the genetic inheritance passed from mother to daughter, tracing female lineages over generations. In turn, Y-DNA investigates the Y chromosome, passed exclusively from father to son, and is essential for mapping male lineages. These studies reveal not only the impressive genetic continuity of Jews over the centuries, but also the mixtures, migrations, and interactions that shaped this people's identity throughout history. They offer a window into the past, connecting the present to ancient Semitic DNA. Regarding DNA samples from ancient Jews, one of the most cited studies currently as an example of DNA from ancient Israelites pertains to the genetic material recovered from the remains of Israelites who lived during the First Temple period. These DNA samples were extracted from bones found at the archaeological site of Kiryat Yearim. Professor Israel Finkelstein led the research, in which the remains of two individuals, a man and a woman, were analyzed. The study revealed that the man belonged to the J2Y DNA haplogroup, while the two identified mitochondrial haplogroups were T1 and H87. It is important for everyone to know that both the DNA of Arabs and ancient and modern Jews predominantly share a genetic fraction known as Neolithic Levant, which I refer to as the predominant fraction of ancient Semitic DNA. This genetic component is widely present among both Arabs, especially the Arabs, and Jews. All peoples of ethnic racial origin from the Middle East, both in the past and present, have as their major fraction of ancestry the component known as Levant Neolithic Farmer. This intriguing component corresponds largely to two ancient genetic groups: the Natufians, who contributed about 33%, and the Neolithic farmers of Anatolia, who represented approximately 35%. These two groups formed the Levant Neolithic Farmer, which is the basis of the ancestry of Semitic peoples, at least in their predominant fraction. Now, speaking about some well-known Jewish groups, in the previous video, I did not detail a specific group: the Sephardic Jews. This group is one of the most famous, especially in Latin America. Many Sephardic Jews migrated to countries like Brazil, Mexico, Argentina, Venezuela, and Colombia, both during the colonial period and in more recent waves of migration. Additionally, Sephardic immigration to the US also occurred in several stages, with a significant presence in cities like New York and Miami, mainly of people coming from Latin America and the Middle East. Let's discuss some information about the DNA of Sephardic Jews, starting with the genetic percentages of ancient Israeli DNA and initially focusing on the paternal lineage, that is, Y DNA, so everyone can better understand the paternal genetics of this group. One particularly relevant study was conducted by geneticist Harry Ostra from the New York University School of Medicine, NYU. Ostra and his team carried out several researches to map the DNA of various Jewish populations, including Sephardic and Ashkenazi Jews. His research helped confirm that Sephardic Jews share a common genetic heritage with other Jewish populations, tracing back to the Middle East. Researchers found that Jews living in Morocco and Algeria showed more European admixture in their genetics than Jews living in Tunisia and Libya. This is likely due to a larger number of Sephardic Jews expelled from the Iberian Peninsula after 1492 and 1497, who settled in these two earlier regions. However, all Sephardic Jewish communities in North Africa exhibited a high degree of endogamy. A study conducted by Inés Nogueiro and others in July 2009 on the Jews of northeast Portugal in the Trás-os-Montes region revealed that paternal lineages consisted of 35.2% typical European lineages, that is, of European origin, R 31.7% and I 3.5%, and 64.8% lineages more characteristic of the Middle East than of Europe, E1, B 18.7%, G 3.5%, J 36.8%, and T 15.8%. Despite a considerable portion of 35.2% European heritage through the paternal line, Sephardic Jews exhibited a predominance of 64.8% genetics coming from the Middle East. This means that paternally, Sephardic Jews are more closely related to other Jewish populations than to non-Jewish Portuguese individuals. Regarding the mitochondrial DNA, mtDNA, of Sephardic Jews, the analysis of these populations, especially from Morocco, Tunisia, Libya, as well as Portugal and the Iberian Peninsula as a whole, was the subject of a more in-depth study in 2008. This study revealed many genetic similarities between Ashkenazi and Sephardic Jews concerning mitochondrial DNA of European origin. Some proposed genetic percentages include 27% European contribution among Moroccan Jews, 43% among Tunisian Jews, and 39.8% among Libyan Jews. In addition, it has been analyzed that the 39.8% mitochondrial DNA of Libyan Jews may be related to a woman carrying the X2E1A1 lineage. Now, speaking about genetic data of ancient Israeli DNA, Sephardic Jews exhibit about 30% to 43% of ancient Israeli DNA in their genome, which is quite similar to what is observed in their Ashkenazi Jewish brothers, who also have similar levels. The Iberian European heritage accounts for 57% of the DNA of Sephardic Jews, coming close to the genetic composition of their Ashkenazi brothers. On the other hand, when addressing the Jews of Ethiopia, known as Beta Israel, or simply Black Jews, it is interesting to note that several genetic studies have been conducted on this population. From a genetic perspective, despite their claims of direct descent from ancient Israelites, it is important to analyze their DNA through scientific research that seeks to confirm whether they truly possess ancient Jewish ancestry, especially from the tribe of Dan, as some assert. Let's start first with uniparental lineages. Genealogical DNA tests allow for the investigation of both paternal and maternal ancestry. According to studies conducted in 2002, haplogroup A is the most common paternal lineage among Ethiopian Jews, carried by about 41% of Beta Israel. This clade is primarily associated with Nilo-Saharan-speaking populations and also with Khoisan peoples. However, the branches carried by Ethiopian Jews are mostly of the variety AY 23865, which formed millennia ago in the Ethiopian highlands. Additionally, about 18% of Ethiopian Jews have lineages from haplogroup EP2XM 35XM2. In Ethiopia, most of these lineages belong to EM329, which has been identified in ancient DNA isolated from an Ethiopian fossil approximately 4,500 years old. These haplotypes are frequent in southwestern Ethiopia, especially among Omotic-speaking populations. The remainder of Beta Israel mostly belongs to haplotypes related to haplogroup EM35JM267, which are commonly associated with Cushitic-speaking populations in northeast Africa. More detailed analyses indicate that haplogroup EM35, carried by Ethiopian Jews, primarily originates from the Horn of Africa rather than from a Levantine origin in the Middle East. A mitochondrial DNA study conducted in 2011 focused on sampling maternal ancestry from 41 Beta Israel revealed that they carried 51.2% of the macro haplogroup L, typically found in Africa. The remainder consisted of Eurasian origin lineages, such as 22% R0, 19.5% M1, 5% W, and 2.5% U. However, no identical haplotype was shared among the Jewish, Arab, and Ethiopian populations, suggesting very limited gene flow between these populations and the possibility of distinct maternal ancestry histories. The maternal ancestral profile of Beta Israel is similar to that of populations from the Ethiopian highlands. Regarding autosomal ancestry, the autosomal DNA of Ethiopian Jews was analyzed in a comprehensive study conducted in 2009, which investigated the genetic affiliations of various African populations. According to clustering analysis, Beta Israel generally clustered with other Cushitic-speaking populations from Ethiopia. Beta Israel has a greater genetic proximity to populations from the Horn of Africa than to any other Jewish populations, including Yemenite Jews. According to a 2020 study, the DNA of Ethiopian Jews is primarily of Eastern African origin and not Semitic, but about 20% of their genetic composition does indeed have Semitic origins from the Middle East, showing similarities to modern Jewish and Arab populations. Despite the presence of Jewish DNA being quite limited among Ethiopian Jews, it is still detected at levels of about 10%, mixing with Arab DNA, as revealed by studies. Thus, the total amounts to about 20% Semitic DNA coming from both Arabs and Jews. In comparison, the Eastern African genetic origin of Beta Israel is much greater and predominant, corresponding to 80% of their genetic composition. I thank everyone who has watched the video so far. A hug to each of you. If you're interested, I can make a third part about ancient Israeli genetics. Leave your comments if you already knew more about the genetics of Sephardic Jews. May God bless you all, and see you soon.